Now I will hand over the next session to moderate by Soumyadipta from Gangeya Student Chapter. Soumyadipta, you can go on.
Thanks, Pallab. Uh, Good afternoon, everyone. Welcome to the first episode of Crosstalk, where we will discuss and listen to the opinions of our renowned personalities upon various subjects and topics. Today's topic is theory and experiment, two sides of the same coin. Now, it's a well-known fact that whenever theories and experimentalists have come together, they have done wonders that have stunned the world. So, let me present my slide here. So all of you can you see my screen?
Not yet.
Okay.
Hello, Athi. Welcome to our meet while Soumyadipta shares. Okay, Soumyadipta, we can see it.
Okay.
Is Kanchan also around?
Should be here, I guess. I don't see him here.
I don't see him either.
Yeah, I don't see Kanchan. He was traveling yesterday for some emergency, but we had a short meeting. Not sure.
Could one of you, Neelan, I don't have his number actually.
Give me a minute. I will just call him.
Yeah,
Okay?
let's check.
While Soumyadipta is getting started, I'll try to call him. Okay.
Sure.
So we can wait a few minutes.
Now, Soumyadipta, I think you get started.
Okay.
I just try to contact Kanchan.
Okay. So our today's topic is theory and experiment, two sides of the same coin. So I just very briefly can present a few examples from history of science where collaborations led to some wonders. In 1943, the famous Luria-Delbruck experiment, we know, where Max Delbruck was a physicist and Salvador Luria was a microbiologist. And they both ultimately demonstrated that in bacteria, genetic mutations arises in the absence of selection. So those are inherent. And it infers that Darwin's theory of natural selection applies to the bacteria and as well as more complex organisms. So it was a groundbreaking work behind which this wonderful collaboration was there. Next, we can say about DNA helic structure discovery in 1953 by the biologist James Watson and as well as the Francis Crick, who was a physicist. So they both, they jointly discovered the double helix structure of DNA using X-ray diffraction techniques and as well as the mathematics of helix transformation. So there are a lot of examples in the history of science where this kind of collaboration led to some great discovery. And I can say that presently in India also, there are some initiatives to promote collaborations and as well as interdisciplinary research, such as the establishment of IISERs and TIFR-TCIS. So to shed more light on this exciting topic, today we have a versatile panel of experts from different parts of India, and let me very briefly introduce the panel to the audience. Welcome Dr. Athi Naganathan. He is presently an associate professor in the Department of Biotechnology at IIT Madras. Welcome Dr. Naganathan.
Thank you.
Thanks. Then we have Dr. Kanchan Garai, who is presently reader in TIFR Hyderabad. We are very happy to get him. Welcome, Dr. Garai.
Unfortunately, Soumyadipta, there must be something wrong. I was unable to get Kanchan on the phone. And I know he was traveling yesterday, so I am completely not aware of what happened. He didn't update us.
Okay, ma'am.
Yeah, I guess we will have to continue nevertheless. And of
course,
many experts in the audience as well. I can see, of course, Bidisha is there. I can see Shumon, right? So I think between us, we will be having enough to discuss.
Right. I think we can obviously proceed. And finally, we have Dr. Sengupta, who is an assistant professor presently in Department of Biological Sciences, IISER Kolkata. Welcome, Dr. Sengupta, and welcome all. Now I don't want to spend more time, and I just want to jump to the discussion. So I would request if one of our panel members can share his or her thoughts about today's topic. So please.
Yeah, Neelanjana, do you want to go ahead?
Yeah, I mean, so basically we had this brief discussion yesterday, and firstly, thank you, Soumyadipta. I think you have launched out very well, and you cited some very nice examples of theory and experiment collaboration. Those that are focused on biology, yes. But if you look at science in general, this has been going on, for ages, right? If not ages, centuries certainly, right? If you go back in time and look at what Newton did and further down, right? I mean, all of these were initially theoretical concepts, right? Laws of motion, even your theory of relativity, so on and so forth, which kind of came to fruition with experiments, right? So broadly, when we were chatting yesterday, we kind of, looked at it, you can categorize these efforts perhaps into A and B, or 1 and 2. One is where, it's theory-inspired experiments, where you actually have a hypothesis inspired by theory and it fits some kind of a very neat mathematical model. And, the description is so intact, it's so beautiful that, you feel that, this must be real. And many a time you will have experiments trying to actually establish it. Of course, there are instances where such a real looking model can also, prove that. We know that Newton's laws of motion work, but until they don't work, until you go into extremely small systems, right? And in the second category, you have, experimental observations, actual observations, which is so frequent in biology. And those phenomena drive theoreticians to come up with mechanisms. in order to explain them. So these are two extreme categories that we think, and my co-panelists and I, we made a WhatsApp group and we were actually going over this. And everybody was busy, but still, Athi, for example, we were trying to look at examples from each category. So in category A, of course, if you look at biophysics, from biology in general, one of the things that you actually said, Darwin's theory of natural selection was actually a theory, though he traveled a lot and stuff like that, and he did not build a mathematical model. Other people did later, but that was actually a hypothesis, a model, right? And there are many more that you cited, the DNA structure, then you have the Lotka-Volterra model for predator-prey, the Hodgkin-Huxley model to explain action potential, so on and so forth.
Right.
But you also have, when you go the other way, where you have an experimental observation, which is like, really exciting. And then you get people to try to, explain that. So a very recent thing that Athi and I were discussing yesterday is this so-called liquid-liquid phase separation, formation of membraneless organelles within cells and so on and so forth, right? So, how does that happen? Now we have a huge number of theoreticians, and no matter where you look, every other person appears to be working on LEPS, right? So that's what we were joking. I don't know which paper to read at a given point in time. But, there are plenty of examples. But I also think, and Athi, let me know what you think, there are many a time where you have two kinds of people talking, right?
Yes.
And it's kind of really, we cannot really bifurcate it into category A or category B, but really it's multiple minds coming together and deciding to look at something.
Yes.
And then you have to, iterate or kind of till you reach mutual convergence of some kind. So Athi actually has, he does both. He does experiments as well as, model building. So in protein biophysics. So I think Athi, you can, or Soumyadipta probably has specific questions.
Yes. Actually, I was also searching Dr. Naganathan's profile. That I saw that he did the both simultaneously, and it seemed to me quite amazing. I mean, and I think it's quite a bit rare also. So that's why I want to ask you, Dr. Naganathan, that how is your experience of integrating these both approaches simultaneously? And what are the advantages? Can you share your journey? I mean, a bit.
Yeah, a journey. Yes, I can share this easier part. But before that, as Neelanjana was also saying, if you think about the actual the scientific problem at hand, and you read many of these research articles back then meaning I'm talking about maybe 20-25 years ago, or even maybe 15 years ago. Right, when you read a research article, particularly done by biologists, right? So biologists, purely biologists, what you will generally have at the end of these research articles are some model of how their observation fits in to the overall scheme of things, right? So generally you will have a diagram. No, I'm not talking about a mathematical model, but it will be some picture, a pictorial representation of how their observation in that particular research article fits into the overall scheme of things. Back then, that's the way it was. But nowadays, it's not like this, right? So predominantly, when you have some experimental observation, what people generally tend to do is that they try to take it up and then they build mathematical models around it with physicists and mathematicians sometimes, and they come up with either stochastic equations or differential equations and try to see, whatever number or whatever quantity they have observed can have an impact, and is that impact really driving the overall change in the say, for example, the organization of the cell, the phenotype, or say even, for example, in liquid phase separation and so on and so forth. So nowadays it's generally not the case where you see specific biologists coming up with some models at the end of the research articles with just giving these cartoons that they make in PowerPoint, right, where you have the circles and these squares and then they come together and then you have this arrow that points to something else that is happening. That's, though you do see them, but nowadays it's kind of frowned upon, because now there are tools available where people can actually take it up and really model them. For example, one interesting example is systems biology, right? So back then, we had lots of genomic data, we had metabolomic data, we had protein-protein interactions, and yeast-2 hybrid systems, and so on and so forth. But how do we kind of integrate them together, right? So, and that has been one of the primary role of theory, really, where it has taken disparate sets of data from different fields, really. And kind of integrated them and showed that, okay, this is the single coherent picture that simultaneously explains the majority of the data. You will always have outliers, you will always have some aspects that are not really, being explained well. So maybe if you have a 70% agreement with the kind of experiments that you have done and the model that is predicting, then I would assume that that's actually a reasonably good model to start with. So, but to improve the model, you will need experiments, and to test the predictions of the model, you will also need experiments. So that you will always need a crosstalk between these two, and I believe majority of us, when we write research articles nowadays, writing a computational article is really difficult, actually, if you think about it, because it's very easy to criticize that, where they say, oh, okay, this is your hypothesis, you're showing this in your simulations or in your theoretical model, okay, but how do I know that this is really happening? Then you will need to calibrate your models against previous observations, that experimental observations that one has, and try to say, okay, since there is these set of observations, we are reproducing all of them, based on which we are predicting some some new aspects associated with the system, like proteins or LLPS or in systems biology related approaches, or as Neelanjana will do in terms of molecular dynamics simulations. So you will need to always have a crosstalk, and I don't see any way out here. So if anyone is doing experiments, I would say, okay, please start working on models and computational side of things, learn coding, and proceed with that. That's what I would say. But I don't want to get too much detail. But in terms of my journey, I had a similar experience where I started off by doing my PhD. I started off doing experiments, and for various reasons, I was kind of attracted to computational models, simple statistical mechanical models, and I moved in there. And I thought maybe that's the best thing to do in life. And then in my postdoctorate, I actually worked entirely on molecular dynamic simulations and coarse-grained simulations of proteins and protein DNA binding. But at the end of about, say, 2 to 3 years of my postdoctorate, I realized that this is going nowhere. And since I had some experimental background, I was finding it difficult to really integrate what the model was predicting versus what experiments observed. So there was no real bridge that can kind of connect these two. So okay, I took it upon myself, among others, right? I'm not the only person. There are 1,000 others doing this, and started doing both experiments and simulations-based approaches. I don't do much of molecular dynamics anymore, but primarily theoretical modeling. But that has reaped dividends, really, where I can see that when I have an experimental observation, I can really explain it at a quantitative level, and take it up further and say that these are the set of reasons why this is happening, and this gives me a collection of predictions from the model, which I can then go back and test it by experiments, and there is always this cycle, right? You go back and forth, one feeds onto the other, and so on and so forth. So it has been a real fun thing to do. Though I find it difficult to convince students to do this modeling and related aspects. So at majority of them, at least the students do not have the background, but at least in some of the courses I teach, I do only assignments which are completely related to mathematical modeling.
Okay. Thanks for such a great insight. And yeah, I quite also, I am a second year PhD student. Just so I feel also this kind of thinking. So yeah, then to Dr. Sengupta, now I can come to you. That you are a theorist. I mean, you do computer simulations and
we do simulations. Yes.
Yes. So now, what do you think about collaborating with an experimentalist? I don't know much about that. So And particularly when do you feel the urge to do a collaboration? What is the driving force? I mean, can you share?
So, from whatever experience I have, I mean, I have a few running collaborations as we speak with different people. Some of them here at IISER, some outside. The first challenge is to come to at least a semi-converged point, because unless you're looking at, a very particular kind of system. I understand proteins well. There are, protein biochemists who understand protein well. So we have some kind of a natural common ground. But generally speaking, the first part where you start speaking the same language, or at least understanding what the other person is talking about and appreciating that person's line of thinking, once that barrier is broken or overcome, it becomes more streamlined. That's what I have seen. I mean, I'm working with a group where they are looking at, fuzzy binding of intrinsically disordered proteins. It's a completely an experimental group. But fortunately, they understand our language to some extent. When we say multi-funnel energy landscape, they know what we are talking about. So to get to that point, unless you know somebody personally and you're talking frequently, getting a collaborator to come to that converging point, I think that is the first barrier. It helps if you're in the same place, but nowadays, we are also seeing that we have probably overestimated the importance of being in the same place. You can also be geographically somewhere else and start talking. It hopefully should not matter. So I think that is the first point. And of course, get people talking. I think, let's say there are two groups collaborating, and you have the PIs who have, who are older people, have their own kind of thoughts. They, they have to teach and so on and so forth. But the students who are working, or the postdocs who are working, they should actually meet and talk more frequently. Okay, so the more you read, or the more you appreciate, and you also have to be creative. You cannot be ultra rigid that, this is the only unique way of looking at this problem. There could be multiple ways, and you have to actually appreciate that there are other possibilities as well. Okay, so that is what I feel. There's also, it's slightly different from the core question that you asked, but as Athi was saying, and since I see Shumon and others as well, can I add something, going back to the previous point about experiments inspired from theory, right? If you look at molecular dynamics, right? I mean, right now there's so many collaborations between molecular dynamics and experiments going on. But if you look at the inception of molecular dynamics back in the mid to late 50s, right? The whole concept was from a theoretical physics point of view, right? So you have an empirical force field. Okay, if you actually, if you have an empirical potential, you derive the forces, right? You get the phase space. Okay, you add thermodynamic controls. This entire thing was a very, an empirical idea, which has actually refined so much over the decades that at this point of time, if you make a prediction based on a very good simulation model, the chances of you agreeing, at least in the sense of trends in certain quantities, with what an experimentalist is showing, the chances of that are very high. That's why nowadays we can actually produce standalone theory simulation papers based on these force fields. So, I mean, it's hand in hand. You cannot just bracket anything in one box and say that's it, right? And nowadays the world is becoming so interdisciplinary that you actually have to look at other people's perspectives. It's a little bit away from off tangent what you asked, but I just felt that is one thing. Shumon, if you have any comments on that, you could also say at any point of time, or anybody else, or Soumyadipta, if you have questions, go ahead.
Yeah, I think at this point, Dr. Vidisha Sinha or Dr. Soumya Chakraborty, they can join us. And okay, so
Is Suman around?
Shuman, are you there?
I can, of course, add. You know, we are old people. We can talk anytime you ask us to. And talk. Also, you are the moderator, Soumyadipta, so you please
Yes.
stop me whenever you want
Yeah,
to. And this is a favorite topic, you know.
Sorry.
Sorry, Athi, go on.
No, Vidisha, go ahead.
Yeah, this is a very favorite topic because you know, I think.
Yeah, and one thing just I want to add that Dr. Sheena, you are an experimentalist here. So I mean,
it's another. But Athi is has seen from both the angles, right? From experiment to theory and theory to experiment. So I think he has already given us that insight.
Right.
But I'll tell you what happens, and I mean, from my own experience, sometimes when you want to understand mechanisms, because biology is complex, you cannot say ye bad gaya to ye hua, you know, this has gone up and this has caused this. That's just, usually that is not enough. That is not the whole story. And if you want to know the underlying design principles or the mechanisms, there is no way out but to try to create it. I think recently somebody had, it was, I think it was Feynman who said, you know, I don't believe something that I can't create. While the, I think Francois Nedlec has recently, I mean, somebody posted it on Twitter that sometimes you don't understand what you create. Of course, that is also there. But so for the complete understanding as an experimentalist, if and when that happens, that a theory can give you the underlying mechanism, that is extremely satisfying. And you know that then perhaps what your experiment says is not incorrect, or not, I would not say incorrect, but not incomplete. You know, that is from where that is one part. And the other part, I think, as an experimentalist and somebody who does microscopy, like we just heard Dipanjan, and it is very important for us to not think of our tools as tools, because these tools come out of deeper understanding of light and matter interaction. So they're not just tools unless you lift those tools as, I mean, think of those tools as absolutely completely research topics, as not just, okay, it's a tool I will pick up and use. Then, so there is again the need for we as a my microscopist, I feel the need of talking to theorists for the even like the in-depth understanding of what I'm using, because only then perhaps I can take the experiment to a regime where it will tell me something new, or it will tell me something which the current techniques can't. And biology has gone hand in hand with the progress, you know, how microscopy has progressed. So again, this is something which, you know, when we train you as biologists in the different places we do, we tell you to think about questions, and then, but what we call tools are actually also questions in themselves. And that is where I think not bracketing anybody, like something that Neelanjana was talking about, is applies even there. And for interdisciplinary fields like this one, bracketing is only going to restrict you to one direction, and you cannot broaden, you cannot you know, even try to ask difficult questions. So I think that's all I wanted to say. Yeah, because after this I will be repeating myself.
So yeah, I have something similar to add here to Neelanjana.
Yes, yeah.
So it's something what Neelanjana just kind of alluded to on liquid-liquid phase separation. So until now, people have been studying, you know, these solid proteins and, you know, the dynamics associated with some loops and residues and so on and so forth, right? So that's what people have been studying. But now the entire field has kind of been turned upside down. Now people are running helter-skelter to identify or come up with models that can explain this beautiful phenomena that they see in terms of liquid-liquid phase separation, really. So, and there is, though you can say that there are several different models, there is no real a single model that can explain everything consistently. So every time people do an experiment, they observe some discrepancy with the model, they go ahead and fine tune the model, and that's what is happening with molecular dynamics simulations currently, right? So people find that, okay, you start off, you have been parameterizing these force fields for well-folded proteins, and it seems like some of these cannot be directly transferable, not all of them, some of these cannot be directly transferable to disordered proteins, right? Which now you see quite a bit of it in biology, where a large chunk of the proteome is made up of disordered proteins, and some of the force field parameters that have been used, and maybe Neelanjana can maybe discuss that more. They cannot be directly transferred to disordered proteins because now the way things have arrived at are quite different. And so now people are now going back and see if they can come up with a self-consistent model that can explain both folded proteins, disordered proteins, and also specific experimental observables that come from, say, NMR or dynamic light scattering, for example. So and you will need this crosstalk. You will need to do this, and this is the only way science can move forward, right? And because there is no way to say that, okay, you tune these parameters and then everything else will fall in place. That's not going to happen anymore. And this LLPS is a great example. Yes, definitely. Neelanjana, maybe you can have
absolutely. This LLPS business, and LLPS is going on for about a decade, I would think. Athi, what do you think, right?
Yeah.
10 to 15. I mean, when I was doing a PhD, it was not hot at all. I don't remember reading any articles.
2009 when Hyman and Brandwein.
Yes. And you know what? On one hand, yes, okay, you have to use the right screwdriver for the right nut. In other words, right now, you know, coming to the very limited domain of discussion on molecular dynamics, you have to know what you're doing. Okay, and if your model is not good enough, you have to be smart enough to actually tune that model. So I cannot assume that the parameters, these are empirical parameters which are designed for folded states. I mean, are they going to work equally well when I'm talking of something which hops conformations very easily? In other words, IDPs, right? So intrinsically disordered proteins. So you have to think on those terms. And nowadays we have, you know, many different kinds of tools to help us incorporate extensive information into models, right? So let's not get into machine learning and so on and so forth, but there are ways in which you can actually autocorrect many of these things. But there was something else I was, you know, I already said that, you know, the domain of simulations itself, you know, the way it started. Okay, back then, I think, you know, the earliest papers, they were just trying to get the structure of water correct, and very, very, you know, I think it was argon and things like that, to the point where you can actually get, you know, thermodynamics and kinetics quite accurately. Okay, well, in many cases within experimental error limits as well. Right, so you know, those are you know, that's a huge success, and it's taken some 6 decades to go forward. And if you excuse my, you know, just moving around from hopping from one thing to the other, coming back to this whole LLPS things, and it's of interest to biologists because you can form these liquid-liquid phase transition is thought to be the reason why you can have membrane-less organelles in, you know, in complex media such as the cytoplasm. Now, this is also related to another phenomena which was observed experimentally, and then there was this huge debate. If you have heard of supercooled water, it's actually a bistable state. You have a high density amorphous state and a low density amorphous state, and, you know, it's a 2 basin thing. You know, you have 2 states. Now, for the last, I think this whole debate between 2 groups, 2 very prominent groups in this domain, there was a debate that raged on for about 10 years. Okay, and 2 theoretical groups. Okay, they started out as friends to what I know, and, you know, there was this huge disagreement between whether you have just a single state of supercooled water or do you have, you know, 2 states. So, and you know, it was resolved in a very strange way. It turned out that there was a coding error by one of the groups. So it is actually a 2-state thing. I'm not going to name the groups here, but I think some of you will know and you can read up about it. that's also the point, it's imperative on whatever we are doing to make sure we are as accurate as possible within the constraints given. Okay. And many a time, if we actually collaborate with somebody, if we collaborate with somebody with a different expertise, many a times our own methodological errors or systematic errors come into the fore. And once we get that, we must recognize that and move ahead. I've seen quite a few people, so stuck up on their own ideas that they refuse to self-correct or refuse to look at something else. I think it doesn't help anybody. Right. So I'm not trying to be negative here, but I'm just trying to say that these are some general principles on which, if people work, then fields themselves can actually move ahead.
Yeah. So I agree with Neelanjana on that. So by protein folding people, at least I can say for them, maybe so, right? So what at least from my perspective, what people have been studying so far are only 30-40 residues, 70 residue proteins. And you talk to a biologist, they will call that as a peptide. But we so-called protein folders would say that's a protein by itself. So and we have been studying this, and I was having a collaboration with a group from Spain, and they were working on like a 320 residue protein, if I'm not mistaken. So, and they were asking me if the model I use actually works on this system. I said, sorry, this model has been fine-tuned, similar to MD simulations and so on and so forth. It's been fine-tuned for smaller systems, and it will not be applicable because the number of states, the phase space will kind of grow exponentially, and I would not be able to simulate. That actually got me thinking, and we, in collaboration, after having discussed with these experimentalists, we were able to come up with a different approximation of the model that now can be applicable to even 500 residue proteins, the model that I'm working on. So it's a statistical mechanical model, but that helped. I mean, if I was in my own world, right? So as Neelanjana was saying, I'd just be there working on these small thingies and how these things move around and so on and so forth. That would not be of help really. So it's very important to open up and embrace the collaboration.
Right. And in that way, I think theory and experiment are, actually they are complementary to each other.
Yes.
Yeah.
Well, one rides on the other, definitely, and I mean they cannot be standalone entities. Where one says something via experiments and another says something via theoretical work or simulation. So they will have to be kind of weathered together. So and that's the collaboration.
Thank you. And so then I have another question that it perturbs me a lot sometimes, that what are the major difficulties, or I should say major challenges during this kind of collaboration, and especially in interdisciplinary collaboration? So what are the major challenges, and how to overcome those difficulties? What do you think? I mean,
so Neelanjana, do you want to take it up? Because you have been collaborating quite a bit. So.
Well, you have been collaborating with yourself, if I can say that. So I just try to say that, it's first coming to the same platform, but then it kind of depends on, for some people, it's easy to collaborate because they have coffee every day and they're probably chatting anyways, right? For some people, it's more difficult because sometimes personalities get in between. Sometimes, the fact that there's a huge domain gap that comes in between. But I feel that if you're flexible and if you're creative, all these factors should not come in between. In fact, if you do not mind, I see my colleague Arnab Gupta here. And I was wondering if Arnab, in a general sense, you could comment on what are the challenges towards interdisciplinary collaboration. Arnab, do you happen to be here? Because I think he's somebody who collaborates quite a bit with chemists and others. Maybe he's not around, but as I said, I think the first thing is to kind of have the big picture in mind. What is it that you're trying to address? Many a times, if you're missing the forest for the trees, if you're looking at, details are important, Okay, if you get your details wrong, that's what the two water groups were doing. One in the west coast of the US, one in the east coast. The devil lies in the details, certainly. But please have a look at the bigger picture. What is it that you're trying to address? And if you have two very seemingly disparate groups trying to look at the bigger picture together, I think, the technical details and the lingo, it takes time. It might take a year, even a couple of years, but eventually you're going to get there. And I think what is, again, I don't want to sound philosophical or anything, it is kind of respecting other people's points of views as well. Right? Maybe I'm harping on and on about the same, I'm just not looking at another point of view which is presented by somebody else. If I did, maybe I would actually improve my entire model. So, I think it's just how you approach it. That's what I tend to think. And in fact, Soumyadipta, your department, your advisor, I think there are good examples of these cross-disciplinary collaborations here in India.
Right.
Okay, and many different places.
Vidisha has.
Vidisha, please.
I thought I would just add and pitch in for Arnav. So, yeah, I mean, these are very important questions. I mean, it's not so easy to collaborate. You're right. So, one of the you should also be aware. I mean, both the parties should know that it is well possible that things don't go in the direction that you were expecting. So, and then that may lead to that, a phasing off of the collaboration. So if you have super planned things in your head and say, this is what I'm going to get and this is what I'm going to achieve and then maybe that's like pushing too much. So you have to keep the balance, and you have to, as you go along, you will understand if is that collaboration going to teach you something. It's not always about cracking a problem. Sometimes, like Neelanjana was pointing out, people who have coffee may have an easier way in their collaborations, and my best collaborators are ones from whom I have learned a lot. And actually the discussion started in a coffee shop, and it would go on in a coffee shop in that pace, I'm saying. While so there are different kinds of collaboration, there are also collaborations where you know exactly what to do together. So maybe in a few months things are coming into shape, but that both these collaborations teach you a lot. So that the other kind of collaborations, which is a slow collaboration through which you are also, to me, very important. Because I mean, there is, again, like sometimes don't we feel that this talk is not in my field? Should I waste the time listening to this talk? You never know what idea comes to your mind when you hear that talk, and much better than talk is talking to people about their own work. So, and that is where I think you can start collaborations. I mean, the more you talk, and as students, I mean, in my own PhD, I think I've learned a lot from my student colleagues, meaning other people in the department, in the lab, especially when you are in a field like biophysics, where you would have to learn a lot of biology, you would have to learn a lot of physics, you would have to learn a lot of chemistry. Best is to talk about their work, and that starts collaborations. You can have collaborations that finally take place much later, and the seed is sown much at an earlier level. I think this forum is a clear example where as soon as you make a platform like a coffee shop, I think I read somewhere that at one point Vienna had a lot of coffee shops, and that led to the scientific community doing really well. And it's so important. You have to come out of your, I think Neelanjana has also been talking about it, you have to come out of your own way of seeing things. And that happens when you're not thinking about work, you're thinking about something else, you're going and chatting, and so I would really want to see that our students and people in the student chapters who are at least in this geographical location, we need to see that energy out there. Inside your labs, you're not visible. So I think I took that topic to some other direction.
No, that's fine. That's fine. That's a good thing to say. I mean, and when people meet and discuss, and when they are from disparate backgrounds, I think it's I know. And if they talk science, that's another issue, right? So if they do talk science, and I'm sure that if the wavelength matches really, and you might have some resonance that will kick in really. And that happens, and that comes with how you. That also comes down to finally how you talk to others, how do you approach others, how do you treat others, and those issues are also there. But let's not get into that. But maybe we can move on to other questions if there are. And so before getting into that, there is this interesting research article, which I'll just type it. I just have this here. So on how or whether a biologist can fix a radio, and I'm sure many of you might have gone through this. It's actually an interesting article, and this is published, I think, in 2000. So it's 2002 in Cancer Cell. Where the author actually opens a radio and says that all these are the different components that you have in this radio, and even if you remove this particular tuning mechanism that you have, the radio will not work. You will not be able to hear what's going on. So many of the biologists, if I am not being demeaning here, I'm just saying many of the works that are done are actually done in isolation, right? They will look at a particular pathway and they will say, okay, these are the observations that you have, and when you mutate this is what happens downstream. But the same thing, I know, if you consider the analogy of a radio, you see that there are so many ways where the functioning of a radio, in this case the radio is the cell, can be perturbed. And you can just pull out a wire somewhere, a resistor somewhere, a capacitor somewhere, and it will not work. And it doesn't mean that every other component is kind of malfunctioning. It's just that that particular region has gone, and that has complicated things. So you need to have this integrated view, and to have this integrated view, you need to really do both, or all the three. I wouldn't say just simulations and theoretical, experiments and theory, but also simulations, and what kind of simulations you do, what kind of theoretical work you do, as Neelanjana was saying, that depends on the scale of the problem that you're looking at, right? If you're looking at the mechanics of mouse motion, for example, you're not going to talk about proteins and how things get transcribed and so on and so forth. You'll be coming up with some equations that describe the motion of the movement of the mice. So the scale of the problem matters, and appropriately you can choose a collaborator who can help you out in that aspect.
Right. Thanks a lot. And you were saying the fact that of time and length scale. So at that point, just it came to my mind that, then theory and experiment collaboration can also help in time or length scale jump.
Yes, definitely. What happens is that an experimentalist, as we started off this discussion, and Neelanjana was saying this. So when experimentalist does something, right, and you will find that none of the theory that's currently existing kind of fits in. So, and it has to kind of be worked out, or when a theoretician predicts something, and maybe in the current scale of experiments, you maybe not be able to observe some of these things. Even, for example, protein folding, people are still unable to simulate very long time scales for even reasonably sized proteins. I'm talking about 100, 150 residue proteins, and that's a real challenge. And you had David Shaw, who is who is entirely working on computers and computer science, and he's a computer science person who came up with his own hardware, and which is called Anton, to solve this problem, a biological problem and a biochemical problem, if I can say, on proteins and how proteins function, fold, and how to simulate them for longer periods. And that has worked wonders, really. So it matters, yes. You would need to have this collaboration. I don't think we can anymore say that they are two different things. I think every paper, I would assume, that has to have some or theory or simulation.
Right. Thanks a lot for such an insightful discussion to all of you.
sure Neelanjana can have more comments on that or others. I think we might be repeating things unless we have new questions.
Exactly.
Yeah, I think others can put their points of view also.
There
We
are
don't
some questions
have
in the
much
chat
time.
box.
Yeah, in a chat box there is a question from Raunak Adhikary. Besides the IISERs, IITs and some specific institutes in India which provide insights to this side of scientific world, right? I mean interdisciplinary, is there any need to revise the curriculum of current education system followed by state universities or institutions so that India can compete at the global stage?
That's a difficult question to answer because I don't think changing the curriculum would help. Finally, it would come down to the interests of the students and the kind of problem that you're working on. I believe that any biologist should take physical chemistry courses, several of them, and whatever it is, maybe Neelanjana can suggest those what they could be. But that's definitely a no-brainer, and without physical chemistry, I don't think it's reasonable to think that we'll be able to really make big strides in biology. And you see, majority of the people here, I mean, I think Neelanjana, your background is chemistry, right?
I've been all over the place. I did my master's in physics initially.
Physics. All right.
I did
So
my PhD in chemical and materials physics at UC Irvine.
So
And
there you go. There you have a live example, and I think Sumit is also chemist background. I think majority of the people I know in biology actually are in the physical chemistry background.
so what I feel missing is one more thing, Athi, if I may add. So when you talk of physical chemistry, I mean, as our director was saying today morning, it's, that gives you a lot of insights, especially when you're looking at mechanisms and so on and so forth. What I find missing, and that kind of really, nowadays I don't get shocked, but I still get surprised. A lot of people who claim to understand physical chemistry have zero idea about statistics. Not zero, but they've probably done probability and statistics at the 12th course. I'm sure they've got good marks and stuff, but they don't have a feeling for statistics. And it amazes me that such people, I think, so I think there is a little bit of a difference between, it's probably a bit abstract, but learning something and appreciating something internally such that now you can take those ideas to a different domain. That is, I don't know what you feel, but I think that's a person dependent thing. Perhaps it comes with experience and depends on how many problems you have you have handled down the line. But I think so coming back to what Raunak was saying, it's missing in state universities. Hey Rosanna. Hey, good to see you. We are just chatting a little bit. We'll wrap up soon.
Dr. Kolkata, I think you are muted.
Hi, yes, I know.
Hey, lovely to see you. Hi.
Hey guys, how are you?
Hi, we are just wrapping up the last session. We'll just get to you in maybe a couple of minutes. Feel free to add, we are discussing theory versus experiment. And, so how do they complement each other and that kind of thing. So as I was saying, so having a background in physical chemistry is always great. But it's important, but I sometimes feel there's some people who don't understand that you have to look at things from a statistical point of view. I have encountered a lot of students who if I tell them to draw a schematic distribution of people's heights, they have zero idea, but they can answer all questions about quantum mechanics. So
So, Neelanjana, if I may add something there, just like you're saying that one has read statistics but not sort of appreciating it, at least when it comes to interdisciplinary sciences, which also has biology, and since the main system of study, unless you look at biology and see that, wow, this is such a beautiful working and so complicated yet working system, if that awe is not coming out of you, then if you think of again biology as something that I will just use, that doesn't bring out the best. So appreciating, and the word I'll borrow that word from Neelanjana, appreciating a subject is extremely important if you are going to use it as one of the interdisciplinary topics. Hello, Rosanna.
Hi, hello.
Hi, Rosanna. So wonderful to see you.
Yes, really nice to see you, Neela. Thanks for inviting me. I'm really happy to be able to talk about our work.
Yeah, so we are almost done, but there is also a question in the chat box from Priti Roy. Can the theory experiment collaboration cost minimize your research problem at the very beginning?
No, I don't know whether we can you can collaborate to start off with, but I think it has to kind of grow into you that that the particular problem that you're doing, or maybe it's a theoretical work that you're doing, needs to have either an experimental component or vice versa. So it has to be natural outcome, and generally nowadays that is the situation. And I think I also see one question before as to how much biology do we need to know to do research in biophysics after masters in physics. A lot of biology, yes. And as Vidisha put it, and you will need to really appreciate biology. I mean, it wouldn't, again, you cannot say that, okay, I've done all these different things and I can start off by working on some biological problem, but you need to really understand how biology works, and how do cells function, and what are the components, how do they kind of come together, and particularly what kind of experimental techniques that people use to actually quantify some of the observations that they make, because they have their own limitations as well. So as a theoretician or as some kind of person who's doing simulations, you should take it with a bag of salt, really, both sides. And not just a grain of salt, really a bag of salt would probably work. And so be cynical, be skeptical of most things in life, and particularly with these biological techniques and approaches, and also with the theoretical models that people have come up with. The moment you are skeptical and cynical, I think you will probably go a long way in trying to understand and even address problems. So how to encourage? There's one more question there. It is a great thing to see that.
You're muted.
How to encourage young researchers to grow collaborative attitude? No, I'm not sure whether we can. It's
a choice. It's something that you naturally grow into. I don't. You learn by example. You learn from it. So one thing is you read a lot and see exactly what you like. And a lot of students kind of make up their mind at a very early stage that, okay, this is what I like. And they're so set in their own minds that they are not willing to venture into different domains. So as Vidisha writes, a lot of exposure, a lot of turnover and, interactions. That's how you grow. Okay, that's how you grow a niche, I think.
Okay, just want to add one. Sorry, Soumyadipta, just one.
Yes. Yeah.
These transitions we have all made. I have also made this transition. The moment you stop thinking that no, I'm a physicist, I'm just using biology as a system. No, that's not good enough. You have to come out of that and think yourself as a scientist. Otherwise, it is very difficult. You will never be able to cross the bridge. We'll all be on this side. That's a very, very personal sort of experience driven.
I agree with Isha. I mean, if you're somebody who's really curious, if you're inherently somebody who's very curious about many different things, then, and if you have adequate exposure, I think you will naturally fall into, it should not be an upward climb. You really like something and your interests kind of propel you to read and expose and interact. So, you'll find your niche yourself. But again, if you kind of put yourself in one bucket and say, I'm a physicist, I'm up in the hierarchy and I will not do biology, or if you start believing in hierarchies, you're done. You will never kind of get into anything.
Right, complete. Yeah, one.
So
Another question is there. So, what usually prompts physicists to shift gears and pursue research in the field of biophysics? So, this is, I think, it's
again,
common.
yeah, it's not something that can be answered. It has, every person is different, and they will have to take it up upon themselves really. And well, Vidisha has, I think, answered. I think Vidisha should have been here. What, Neelanjana? So next time.
Yeah, I mean,
I'm
she
actually quite there. Except I don't.
was there. She is somehow,
Okay. Yeah. I have been there with you guys. I mean,
yeah, she's there.
told them that if somebody doesn't turn up, I'm there.
Yes. So and finally, it's the beauty. I mean, you should see that, and that automatically would drive you to address some question. I mean, you have to have the curiosity of a kid, really. I mean, and you would see that they are constantly asking you questions where, what is this? Why is this happening? And that curiosity generally dies down, and that's, some people believe that's because of the educational system that we have, where we are kind of streamlined and straitjacketed in a certain manner, and that there are certain ways of learning and reading things and so on and so forth. And but you would see that kids are quite adept at solving puzzles and asking questions and thinking through problems. It kind of dies down with age. But hopefully, if you are able to keep up that particular curiosity, as Neelanjana said, and also Birisha said, I'm sure that you will be able to move across fields and try to address fundamental questions.
So, ultimately, we should do something which is curiosity driven.
Yes.
So, I think,
there's no other way you can do research.
right.
You have to be curious enough, and you have to be driven enough, and you will have to work hard and to get to reach those heights and scale those peaks. It cannot just come by the fact that you might have the most brilliant mind in the world, but if you're not willing to put in that effort, it's not going to take you anywhere. So,
Yeah. Soumyadipta, we are already over time.
Yeah, we are running out of time. So, thanks again to all of you for this kind of fruitful, wonderful discussion. I liked it very much, and I think all of us liked it. Yeah. So, thanks again.
Thank you.
Thanks, Soumyadipta. Yeah, so not only you, I think all of us have enjoyed this great session.